Video, we are going to take a look at the male reproductive system. So this system is a little bit different than a lot of the other systems which are responsible for maintaining homeostasis. However, this system is going to be important in regulating the passage of genes from one generation to the next. So we're going to look at the reproductive system of males in this video and in females in the next video. So the male reproductive system is going to consist of the testes and the external genitalia. We'll go into more detail about some of the internal structures as well. Notice that you have a prepuce or foreskin that cover the penis in individuals when they are born. Many people, however, undergo circumcision, which are going to remove that foreskin. Removal of the foreskin leads to the proper viewing or clear viewing of the glans penis. This is going to be the part of the penis that is going to have the most sensation associated with it. The scrotum is going to be where the testes are found. What's important about the testes is that they actually allow the sperm to start being produced at a temperature that's lower than the body's core temperature. So it's about 5 degrees Celsius lower than core bodyo, which is going to be essential for spermatogenesis, the process of forming sperm. So externally, you can see that the scrotum is bilateral, so you have right and left sides. Separating the two sides is going to be a structure that is known as the raphe. The raphe coincides with the scrotal septum that you would see internally to that structure. On the outside of the scrotum, you have dartos muscles. These are going to be responsible for moving the scrotum closer to the body or farther away, like tightening it. And the cremasters bring them up into the body to help regulate the temperature. The testes themselves are going to lie deep to the cremaster muscles. They will have the epididymis lying superiorly on them. And then you have the pampiform plexus, which is going to be a small network or a network of small veins that drains blood from the testicles and epididymis. It acts as a countercurrent heat exchanger, helping to cool the arterial blood and help maintain that lower body temperature. The spermatic cord is going to be surrounded by the pampiform plexus. This is going to be the actual structure that carries the sperm from the testes ultimately out of the body. If we look at the testes, the testes are the male gonads in the primary reproductive system. So they're going to be responsible for producing sperm as well as androgens such as testosterone. And they function throughout the male lifespan. So each testes is going to be surrounded by 2 connective tissue layers. You have the tunica vaginalis. This is the outer serous membrane with parietal and visceral layers. And you have the tunica albuginea. This is a tough, dense connective tissue layer that covers the testes themselves. The tunica albuginea forms the internal septa that ultimately divide the testes into several lobules. So you can see the seminiferous lobules here. They're actually much larger in this image than they would be in real life because you have somewhere between 300 to 400 of the lobules in real life. So each lobule contains the seminiferous tubules. This is where the sperm is being produced, and the sperm is ultimately going to be produced here and then move on into the next phase of their journey. So if we take a closer look at the seminiferous tubules, you can see that they have a structure with a lumen in the middle. That is where the sperm is ultimately going to go as it develops. On the outside, you have Sertoli cells that are spread out through them. They have branches. These are a type of supporting cell also called a sustentacular cell or sustenocyte. They are found in epithelial tissue. Sertoli cells are going to secrete a signaling molecule that helps the sperm production and controls whether the germ cells are going to divide or not. The spermatogonia are going to be the cells that are the germ cells of the sperm. So they are going to give rise to primary spermatocytes, which give rise to secondary spermatocytes. You don't see them clearly in this image. Ultimately giving rise to the spermatotids that will eventually grow a flagella and be the structure that forms the sperm or the spermatozoa. You also will have Leydig cells. These are found in the testicular interstitium. They are going to be producing androgens such as testosterone and will help to stimulate the male development. They are going to be stimulated by luteinizing hormone and will convert the cholesterol into the steroids. So the process of spermatogenesis is what actually forms the sperm. So you start with the spermatogonium, which is your germ cell. It can divide via mitosis, giving rise to a primary spermatocyte and also replenishing its population. The primary spermatocyte is going to undergo meiosis 1. So this is going to result in the separation of the homologous pairs, resulting in a haploid cell that still has sister chromatids present. So these are your secondary spermatocytes. They undergo meiosis 2, which is going to separate the sister chromatids. So now you have 4 spermatid cells that each have their single, so sister chromatids are no longer present, single haploid genome, unreplicated, which will go through the process spermiogenesis, so not for their dividing, but it is a metamorphic process that is going to result in forming the spermatozoa, which is the structure that we know of when we think of sperm. The sperm structure itself is going to be designed for movement and for penetration of the egg. It's much smaller than most cells in the body and is several thousands, tens of thousands times smaller than the female gamete. Much more sperm is going to be produced than oocytes. So you have about 100 to 300 million sperm produced each day, compared to females typically ovulating only 1 oocyte per month on average. Sperm have 3 distinct regions, so they have a distinctive head, midpiece, and tail. At the end of the tail, you can actually divide that out into an end piece as well. The head is going to consist primarily of the nucleus, as well as a storage site that's known as the acrosome that is going to contain digestive enzymes that are going to be responsible for helping penetrate the egg. The centriole is going to be the piece that basically connects the midpiece and the head. It is going to be what the flagella is ultimately connected to. The midpiece is going to contain a lot of mitochondria, since the tail is going to be involved in movement. There's going to expend a lot of energy and therefore needs a lot of ATP to help power the flagellum. The flagella is going to extend from the midpiece. You will see that within the flagella itself, you have an axial filament that extends both within the tail and all the way up to the midpiece. So the axial filament is what connects to the centriole and what is going to be responsible for the movement of the sperm. All right, so we've got our sperm being formed in the seminiferous tubules. From the seminiferous tubules, they're going to move into the straight tubules and ultimately go to the then go to the rete testes, entering into the efferent ductule, where they'll then go into the epididymis head, move along to the epididymis body, meeting the epididymis tail, and eventually reaching the ductus deferens, which is going to go up to the spermatic cord, which is covered by the cremaster tail. So the sperm is going to take an average of 12 days for it to move along the epididymis. As it is moved through there, it is going to be moved by contraction of smooth muscles lining the tubes. And as they line there, they're going to go through that spermiogenesis and, or really not go spermiogenesis, but further develop to help facilitate their movement and ability to fertilize the egg. The mature sperm will get stored in the tail of the epididymis until ejaculation occurs. The vas deferens is going to connect to the urethra via the ejaculatory duct. You also have the seminal vesicle that's going to mix with the contents of the vas deferens in the ejaculatory duct, where it'll connect to the prostatic urethra. You also have, then enter the membranous epirethia. Once you've reached the spongy urethra, you have additional gland secretions from the bulbourethral gland that is going to help move the sperm to the exit of the penis via the urethral opening. If we look at a cross-section of the penis, you'll notice that you have a corpus cavernosum that is going to surround a large portion of the penis that makes up the body of it. Surrounding within the middle of them, you have two arteries that are known as cavernosal arteries. And then below, you have the corpus spongiosum, where the spongy urethra is traveling. On the superior layer of it, you have the deep dorsal vein that is going to have branches known as penile venules around them. The amount of blood that is present here is going to determine whether the penis is flaccid or erect. So in the flaccid view, you can see that the deep dorsal vein is open and blood is able to flow through it. In the erect view, you'll see that this collapses, which is going to trap blood in the corpus cavernosum. The cavernosal arteries are also going to dilate, so notice that their diameter has increased, so you have more blood in there, causing the erect the penis and the compression of the penile veins and venules that ultimately maintain that erection. Testosterone production is going to be regulated by the hypothalamus. So it releases gonadotropin releasing hormone, stimulates the anterior pituitary to release follicle stimulating hormone and luteinizing hormone. So the follicle stimulating hormone and the luteinizing hormone are going to stimulate distinct portions of the male reproductive cells. Luteinizing hormone stimulating the Leydig cells, causing them to release testosterone, while the follicle stimulating hormone is going to stimulate Sertoli cells. This causes the release of ABP. The ABP is going to, which is androgen binding protein, is going to bind to testosterone, androgen, so that keeps the testosterone levels high. Negative feedback is going to feedback to the anterior pituitary, inhibiting further release of FSH. This happens because inhibin that is produced from the Sertoli cells is going to help turn that portion off. Testosterone negatively feeds back on both the hypothalamus and pituitary, inhibiting further release of the GnRH, FSH, and LH. So we have the inhibin being produced here by the Sertoli cells, and the testosterone obviously being produced by the Leydig cells. All right, that does it for the male reproductive system. Next video, we'll take a look at the female reproductive